United States, in Germany, in some life experience. From the International Space Station, but yet we still have these buildings that you have to go into to find out what ails you and what's the treatment. That's old-fashioned. We can do a much better job of bringing healthcare to you wherever you are. Decrease overcrowded hospitals, decrease barriers to getting healthcare in the first place. I think it's so critical to highlight health inequities that exist in this world, and how we have to do a better job with technology to bring healthcare to everybody. Because if COVID taught us one thing, and I think it taught us a lot of things, what affects one person in this world can end up affecting you. So we have to do a much better job of improving everybody's healthcare. And I think sensors, implantable sensors, nanotechnology might be the answer that I'll talk about. I'm going to talk in detail about medical devices that fail. Do you know we don't have one medical device that is 100% successful? There are failure rates for every single medical device that we use in the body. That's a problem. Overdependency on drugs to fix everything. We have talked already a great deal about antibiotic-resistant bacteria. We created new strains of bacteria from our thought process that drugs can fix all of your problems. In the US, we have an opioid epidemic. People have healthcare problems. They go in for surgery. They get painkillers, opioids prescribed to them. And then when they're finished, the prescription runs up, but they're addicted to the opioids. So they go to the illegal drug market to get opioids. This idea that you go in for healthcare and the solution is for a prescription has to change. We have to be much more intelligent about our medicine. We tend to treat every patient the same, yet everybody has slightly different immune systems. Our medicine can be much more tailored to your particular immune system, your particular health problem. And this is also one of my favorites. We tend to react to healthcare problems and not predict, right? We were talking about a bridge outside of Boston that recently was failing. Fortunately, we had engineers, we had scientists figure out that that bridge was about to fail before it failed. Do we do that in healthcare? We wait for your body to fail, and then we find a solution. That is totally backwards. So we need to do a better job with predictive medicine rather than reactionary medicine. And I could go on and on. And I do want to highlight, because an underlying message to every time that I talk, you can contribute to these problems. I think it's amazing when I look back at my career, We started nanotechnology, nanomedicine in 1990. How many of you were alive in 1990? You don't have to raise your hand. People were not talking about nanotechnology. Nobody even knew what the word meant back then. In my career, we've been able to go through a series of steps with all the tribute going to the students, the startup companies, the CEOs, the investors, and now we have implants that are in humans. That is a difference that you can make if you want to go down that path. And I think me, as well as you two, living proof that we can bring things to humans. It doesn't always just have to be a lab bench experiment. Okay, should I see if this works? I think you're doing that, right? Okay, so I can say next slide and then that'll work. So I don't want to talk too much about this because if you've been following healthcare, you know what a problem antibiotic-resistant bacteria is. Let me just put it into perspective. The US Centers for Disease Control has predicted this will be worse than COVID. So you thought COVID was bad. Wait until we really see the effects of antibiotic-resistant bacteria. MRSA, one of the most common antibiotic-resistant bacteria. It's not the only one. There are others. But look at some of the statistics. The Centers for Disease Control has predicted by 2050, one person every 3 seconds will die from an antibiotic-resistant infection. One person every 3 seconds. It's crazy. Also, they've predicted more people will die from antibiotic-resistant bacteria than all cancers combined. Not to criticize cancer research, but we've been doing cancer research for a long time. How much funding goes into antibiotic-resistant bacteria research? It pales in comparison. We have to acknowledge this is a problem, and I think part of the problem, why we're not seeing this elevated to the levels it should, is unfortunately a lot of people in the healthcare industry treat this as the dirty secret. The surgeon says, oh, I never get an infected implant. Well, somebody's getting an infected implant because the statistics are showing an infected implant. So we have to just acknowledge bacteria will be present if you get surgery. Bacteria is present in the environment. So if you have a scratch on your hand, you might get an infection. We have to acknowledge these things in order to come up with potential solutions. So of course, one of the things, and next slide, please. One of the things that we have been very interested in looking at is nanotechnology, the science of the small. And I can tell you back in my PhD thesis, I had the simple hypothesis that a nanomaterial would be better integrated into the body, could reduce infection, reduce chronic inflammation better than what we're implanting today. And the hypothesis was based on that our bodies are nanomaterials. If you look at bone under a high-power scanning electron microscope, you will find nanoparticles. If you look at cartilage, if you look at vascular tissue, proteins that are present in those tissues are nano-dimensional, nano-structured. So it made perfect sense to me. Use nanomaterials. I will tell you that on my PhD thesis committee, thankfully, somebody argued with me. And they said, mimicking nature is not good enough. You have to have a deeper reason why to use nanomaterials. So I'm going to present to you some clinical data. We've been fortunate enough to have one of our, actually 3 of our implants make it into humans that are showing no cases of failure, no cases of infection, no cases of chronic inflammation over the past 5 years. And in fact, if you add up the patients, it's about 30,000 patients in the United States. These materials are only approved by the US FDA. We're now trying to go to other regulatory agencies and get them approved. But every single one of these implants has succeeded in performing its function so far, knock on wood. But I also want to share with you the fundamental science, not just the explanation that nanomaterials mimic the body, but what is the deeper science behind this? So if you go to the next slide, you'll actually see 3 of our materials. So this is the biggest one, the most implanted. This is formed when I was an assistant professor at Purdue, when all the senior professors were saying, don't start companies, it won't help you get tenure, but I did it anyway. We formed a company called NanoV. So it's VIS, like vis-a-vis, life to nano. We took implants off the shelf. We never develop our own implants. We only change the surface texture. And we do that on purpose. It's a quicker FDA approval process if you're just changing surface roughness compared to developing new chemistries. And we want to help patients now. There's a lot of great research with new chemistries that I'm sure will have a positive impact, but it's probably going to take 10 years, 15 years for those materials to get approved and be inserted in humans. So we wanted something quick. Plus, we had funding from National Science Foundation, National Institutes of Health that were looking for success stories. So they needed things to be done pretty quickly. Okay, thanks. So these are called pedicle screws. Obviously, this is a spine. If you have osteoporosis, your discs might collapse. If you get into a car accident, obviously something could happen. So these screws are placed in patients to fix the spine and return, normal function to a patient. These are titanium. So I'll show you in a moment what the surface looks like, but it's a titanium-based nanotexturing process. We have other devices. This is silicon nitride, different chemistry, slightly different type of device, but same idea, put a nanotexture on that material. And again, in about 4,000 patients, we have seen no cases of failure over the past 5 years. The third example that I can give you, again, yet another implant situation. This is in the foot, obviously, and it uses calcium phosphate, another chemistry. We create nanotextures of calcium phosphate. So 3 different chemistries, titanium, silicon nitride, and calcium phosphate, 3 different types of medical devices, same idea, put a nanotexture on it, and we've seen no failure among all 3 of these. So we're incredibly excited, obviously, about this. The other thing that is brutally honest about this field is you can go on that website and you can see how well an implant does. The FDA runs a very open process where if you're an orthopedic surgeon and you have a failed implant, you have to report it on this website. So as a patient, if you ever need an implant, if you already have one, it might be too late. But if you ever need an implant, go to this website. Type in the company, type in the type of implant, and see for yourself. Is it succeeding or is it failing? So like my kids are addicted to social media, I'm addicted to this website. I go on this website far too often to look up how these materials are performing. So they're doing well so far. I hope it continues, and we'll see. Did I do it? Okay. So just real quick, here's what the surface looks like. This is an anodization process. So it forms what we call nanotubes. Some people call this nanopores. Whatever you want to call it, it's a nanotexture. That specific nanotexture is not an accident. And this is where the fundamental science gets into play. We have an exact equation. We have an exact science for why we create those features to be the size and geometry that they are. But before I get to that, I just wanted to announce, because I'm super excited, that we formed a partnership with Medtronic. Medtronic is one of the major implant companies in the world. They are using this texture of titanium on their own device. So this is another type of spinal device. It has polyether ether ketone peak as the base, and then the structure of the surface is titanium that has been anodized to have that kind of nano feature. And this is what I love about science. So we do all this hard work, right? And then we form collaborations with some of the best companies in the world. They give it to their marketing people, and their marketing people come up with language that's on the package, right? To try to sell the material to orthopedic surgeons. So this is what's crazy to me. Somebody in the marketing department of Medtronic counted all of these nanotubes on the surface of their device, and they came up with 109 billion nanotubes. And then, of course, the marketing team said, well, That's 109 billion opportunities to interact. Emphasize, because having spent my life in nanotechnology, I often heard in the early days, nanotechnology is hype. It will never do anything beneficial to society, let alone health. Nanotechnology, we've been doing that since we've been making organic chemicals in the lab using organic chemistry methods. For a cell to form those charge connections and attach physiochemically to the surface. The first thing to attach is actually water. And it sounds simple, water, but water can absorb in different ways. You can have a hydrophilic surface, you can have a hydrophobic surface, you can have hydrophobicity, surface energy, which can change what proteins absorb from your body, which will change cell behavior. Okay, couple of quick examples. In your body, you have great proteins, not surprisingly. You have one called mucin. Mucin reduces they have a charge. Incidentally, their charge happens to be similar to mucin. So we kind of luck out here, serendipitously. So we develop our implant surface to have the charge that matches vitronectin and fibronectin. We can get more of those proteins to absorb, and we get more bone cells to attach. Guess what IgG has? Just like mucin and vitronectin and fibronectin. A charge. So you can develop, we'll get this right one of these times, right? It has a charge, but this time develop an opposite charge so that IgG does not adsorb, and you can reduce inflammation. So you can see very easily where this idea could be taken infinite different ways. But we're engineers. I'm an engineer, scientist. So one thing we like to do is we like to put equations to this. So I wanted to show you a quick, if you could go to the next slide. I wanted to show you a quick video. So there's two videos here. I don't know if you can click on them, or if you just advance, they should play automatically, I think. There are two titanium structures. One, surface roughness, nanoscale roughness, was created to be hydrophilic. And you can see that water drop spread pretty easily. This was a project we had with Apple, by the way. So this is a 3D printed iWatch cover. They also wanted us to make cell phone protection covers, so out of titanium. So we did that, but we put a different texture on it to make it hydrophobic, so that if you sweat, or you don't want bacteria to attach to your implant, all of these different things, they wanted to know, could we change the roughness to make it hydrophobic? So if you click on or advance the slide, it should play, showing you same chemistry. Both of these are titanium, but this one has a different surface nanotexture, and it's hydrophobic. So the idea is you don't have to change chemistry to change surface energy. So here's hydrophobic. Obviously, the water drop is balling up. You now have the ability to make anything going from hydrophobic to hydrophilic without changing chemistry, but changing surface roughness. That is all we're doing. So if you go to the next slide, you'll see, how this can become a little more complicated, right? Not as easy as what I'm describing. I'm going to give you the 6-step procedure where you can do this. And this is what's fun for me, because if I am totally a business person, 100% business person, I am not showing this to you, because this is our secret. But I'm a professor too. So I want to educate people and let people know this has been our experience of what we've been doing. So you can either do this in your own lab, at home, or give us your implant. Step 1. Okay, so you don't have to do step 1, you can do your own. But if you give it to us, what we want to do is we want to figure out what is the surface energy of your material. We do that by contact angles. Just showed you a couple of them. Here's a couple more. You basically put different liquids on a surface, polar, nonpolar, etc. You measure the angle, plug it into step 3, the Owens-Wendt theory, and you can calculate surface energy of what your implant is now. So if we go to the next slide, this is where it gets complicated. Step 4, we developed an equation that seems to be working. This equation basically tells you what roughness to put on your material to make it successful. So we just calculated your surface energy. That goes in right here. We also know what surface energy it needs to be. Through our 20 years, however long we've been doing this, we have determined for orthopedic implants, if you have a surface energy of 42.5 millinewtons per meter, you will absorb proteins that reduce bacteria attachment and proteins that promote bone cell function. So those proteins have an energy around 42.5. So plug that. Follow this. That roughness is 60.5 nanometers. Guess what that roughness is? That roughness are those nanotubes, that picture I showed you, create titanium to have a roughness of 60.5 nanometers. And that's exactly why we're doing this. Because our equation told us this is the right roughness to give us the right surface energy to eliminate infection and promote bone growth. So step 5, estimate the roughness. I just talked about that. Step 6 is also a hard step. How are you going to do it? How do you create that roughness? And there's, over 60, 70 different ways you can create a nanotexture on your implant. This equation only works if you're not changing chemistry. So in this example, it's TiO2, the oxide that forms on titanium. We don't change chemistry, we only change roughness, and this works, at least in our experiments, it has worked. So you can choose many different processes to create an appropriate roughness. So next slide. We have a term, of course, for this. The business people that I work with said, well, why don't we call this nano-optimized? If you work with a company, if you work with somebody else, we have a trademark that we can give you called nano-op. Your material has been nano-optimized. And it doesn't have to be for implants. It can be for many other types of applications as well. So we've worked with maybe 30 different companies so far and have given them this trademark to show that we followed this equation and we've optimized their surface for a particular biological reaction. So next slide, please. So real quick, how did we do this? I don't know if you're really familiar with anodization, but on the next slide, just a couple of details about anodization. On the next slide, please. This is the cartoon drawing, right? Some people do this in general chemistry courses as an undergraduate. You have an electrolyte solution. We use hydrofluoric acid. I know hydrofluoric acid is not the safest acid to be using, but it works for us. And the FDA approved us using hydrofluoric acid in this process. Here's your implant, platinum cathode, titanium serves as an anode, apply a voltage, 20 volts for 20 minutes. And as you saw, and you can see it again on the next slide, please, you can create screws or titanium in any geometry to have these nano tubes. So that's the process that we followed. We chose this because it's easy, it's quick, it's inexpensive, and the orthopedic implant industry is familiar with this anodization process. They do it all the time with different chemicals, and they don't get this roughness for passivating the surface of titanium. So if you go to the next slide, please. So I'm done. I still have to pinch myself that I remember so distinctly when my first PhD student on this project at Purdue University, Chang Yao, had the beaker, had the alligator clips, had the titanium, had the platinum, applied a voltage, and we saw that this could create a nanotexture. Our in vitro experiments showed that it worked. Our animal studies showed that it worked. And we then formed a company. We had a brave investor back in 2006 who gave us some money to actually explore this. And here we are, 30,000 some patients later, feeling like I made a difference. It could not be any more rewarding than this. So this is from San Diego. I think that I could have taken this picture for countless places in Mexico that have beautiful palm trees, beautiful beach. And I seriously have thought about retiring and just calling it quits, maybe write a novel or something, I don't know. But we're not done, right? So as scientists, it's maybe a little disappointing. We're never happy. We are always trying to improve things. So I asked the question, what else can we do with this idea? How else can we improve medical devices? Because it's not good enough just to eliminate infection in an orthopedic implant. We have so many other implants that could this be used for. So on the next slide, we basically have been using this for other tissues. Some of these are in the FDA approval process, but none of these have been approved by the FDA. So next slide, you can see another tissue that's very close to my heart because 2 of my 3 daughters tore a ligament in their knee playing football/soccer. It is the #1 adolescent female sports injury. Has anyone torn a ligament and received a biomaterial? Please take care of your ligaments and tendons because the process to repair them is very archaic, I would say. Actually, a lot of orthopedics is almost like slightly better than carpentry, I think. So what they do, and they tore the ACL, if you're familiar with the anatomy of the knee, they'll take a tendon from somewhere else in the body. Or maybe they'll take a cadaver tendon. Or it could be a synthetic tendon, although there really aren't a lot of good synthetic materials for tendon repair. They basically take it from somewhere else and they screw it into bone. Suture it back up. About 6 months later, you can now go back to playing soccer. And unfortunately, I think if you play aggressive sports or high quality sports, it'll be about 7 to 10 years before you tear it again. Because all you're doing is pushing the tendon into bone. That's not mechanically how it occurs naturally. Naturally, tendons grow off of bone. So we've been using nanotextures on materials that another company, DePuy, which is part of Johnson & Johnson, we make some of these tendon fixation devices. We put a nanotexture on there. This is a sheep study where we've seen those nanotextures in the supraspinatus tendon, the shoulder of the sheep, can regrow and physically attach to the implant. So nanotextures, we believe, can be used in orthopedic soft tissue repair. Next slide, please. Stents. So can this be used for stents? We think yes. So obviously we all know stents are put into blood vessels that have become clogged or partially clogged. They prop the blood vessel open. One of the big problems with bare metal stents is the metal is exposed to your immune cells, IgG absorbs, macrophages, monocytes come along, form thrombus, form scar tissue, and that reclogs the artery you're trying to open. So you get nowhere. So nanotexturing of stents, if you go to the next slide, you'll see some fluorescently stained endothelial cells. If you can get endothelial cells to cover the stent, that can keep immune cells from recognizing the stent as foreign. So this increased endothelialization is the goal of many researchers. So here's today's stent. Look at all those bare metal spots. That's where monocytes, macrophages recognize this, begin to form the scar tissue. Make smaller, even micron patterns work. The arrow indicates the pattern of the nano feature, but go all the way down to 75 nanometers and you have a pretty good coverage of that surface. So we've seen this is in vitro, but we've also seen in vivo nanotexturing of stents can also help you improve that medical device performance. Next slide, please. Diabetes. So this is a field that there has been a lot of progress, as you probably know, over the past year. And what I mean by progress are the patches that people can wear that measure your blood sugar and alert you if your blood sugar is out of range. You can put your cell phone up to those patches, you can read, it's a real sensor. However, there's controversy for everything, right? There's controversy whether those sensors are reading the correct sugar content in your blood. And the reason for that is for any sensor you put in your body, whether it goes through the skin or it's completely internal, you have to have blood vessels grow on the surface. Because if you don't, you might not be measuring the right amount of sugar in your blood. So getting blood vessels to grow next to an implant is hard. We think nanotechnology could work, but I wanted to share with you a different idea, cell encapsulation. So this is a huge field where people are making micron spheres. They put islet cells, they put stem cells in the middle of these spheres. They inject them into your body or put them subcutaneous, whatever, and those cells are stimulated to produce insulin whenever your blood sugar gets high, right, to help metabolize, insulin helping metabolize that sugar. So if you go to the next slide, you can see some of the issues, at least that I have, with these cell encapsulation devices. A lot of them are polymers. In general, in my research, we have seen polymers initiate inflammation more than metals. And maybe this has to do, I'm sure it has to do with surface energy, increased hydrophobicity for a lot of these polymers compared to metals. So a lot of the pores that are in these polymer membranes, polymer encapsulation devices, get clogged. So insulin can't come out and the cells die. We don't want that, right? So we are proposing on the next slide to use metals. Here's our metals. That same anodization process I talked about before can not only be used to create these, but in a cross section can be used to create these. So here's your cells, here's the membrane wall, and there are nanopores, nanotubes of very specific dimensions that don't become clogged. You can change the shape of these to allow insulin to go out, to keep other proteins or growth factors from coming in that might damage the cells, but they stay open because of their optimal surface energy that reduces inflammation. So we've been doing some of this study with titanium, stainless steel, some other capsules. On the next slide, you can see some of our results. This is a company, by the way, called NanoVault. We're opening up the vault for cell encapsulation technologies. This is just showing you, you can change the size of the pores to allow insulin to be released, but keep things like IgG from coming in. And you have great control over the size of these pores based on that anodization process. This is for titanium, 10 micron thickness membranes that were anodized and showing the release of IgG or release of insulin. On the next slide, it's basically the same result for stainless steel. So this is versatile. You can do this with many different types of chemistries. Next slide. We can also nanotexture the membrane to reduce macrophage function. So this is showing a further nanotexturing of these membrane surfaces, and here's the number of macrophages that are attaching to a nanotextured, nanoporous titanium or nanotextured, nanoporous stainless steel. So you can have an added advantage of reducing macrophage attachment on the surface. And if there's no macrophages, there isn't going to be any inflammatory tissue. So next slide, I think, is the animal study. So here's a rat subcutaneous implantation. And remember what I said before, you need blood vessels to grow up to these devices to keep those cells alive and provide a mechanism by which insulin can systemically go throughout your body. This is our device, nanoporous metallic membranes. This is titanium. You clearly see a lot of blood vessels, right? This is a polymer capsule, cell encapsulation. It's called Viasite, produced by a company. It's being used commercially, not that many blood vessels. So you have an increased problem with this kind of a device and keeping those cells alive. Okay, next slide, please. Cardiovascular tissue. This one gets a little bit at home for me. So heart attack. So if you go to the next slide, I'm sure you're aware when you have a heart attack, part of your heart dies. Now, if it's not a big heart attack or a big problem, a cardiovascular surgeon probably will not do anything. You'll be prescribed blood thinners, stay off the McDonald's, all the other things, but probably no surgery. Well, unfortunately, when part of your heart dies, that means the rest of your heart is working harder. And that means it's not going to last as long. So we and many people are developing cardiac patches, materials that you can either inject, will attach to where the heart attack damaged part of the heart is, or it's a patch that you could go in and try to suture onto the heart to make that happen. So if you go to the next slide, this is our nanomaterial. Here too, we are seeing nanomaterials are helpful. We're using a graphene nanoribbon. They're conductive. Your heart tissue is conductive. That nanotexture we can control. We can make it a certain surface energy to attract proteins that help cardiac myocytes, the cells that beat in your heart, beat. So on the next slide, you can see, if you press it one more time, I think this video should play. This is the material with cardiac myocytes. So, other people have demonstrated this too. You can have a beating heart in culture. We find that this beating occurs earlier when using these graphene nanoparticles that are conductive and have the right surface energy. So next slide. Bladder. Don't everybody run to the restroom. Bladder is another tissue we have been very focused in, and it has to do with bladder cancer. So again, if you have bladder cancer, the solution is not optimal. Probably the tumor, the rest of the bladder gets sutured together. This has an impact on you for the rest of your life. So we are developing another sort of patch, biomaterial, that can regrow the bladder where the tumor was to keep it at the correct anatomical size. So we're using nanotechnology in this too. If you go to the next slide. PGA, I'm sure you all have heard of polyglycolic acid, biodegradable, FDA approved, easy way to create nanotextures on polyglycolic acid, soak it in sodium hydroxide. And you can go from a conventional smooth PGA surface to one that has submicron features to one that has nanotextured features. On the next slide, we put this into rats, their bladder. We resected part of the bladder, and then we inserted this patch. This is the nano textured patch. This is where the urine would be. This is so hard to regenerate in your body. If you do not regenerate the urothelium, you're going to have a lot of smooth muscle damage because the urine is going to be able to go into the smooth muscle and kill those cells. So we got perfect reestablishment of the urothelium. Here is what smooth PGA does. There is no urothelium. It's scattered. It's a mess, so that urine can damage the bladder tissue. So next slide. Nervous system. Stroke, we have been focused on. Again, a clinical problem that doesn't have very good solutions. And we think maybe nanotechnology, nanomaterials might be able to help in this organ system too. So next slide shows probes. You're probably familiar with these. They were developed a long time ago. They basically are inserted. You drill a little hole in the skull, put them into the brain, apply a voltage to jumpstart or reactivate neurons that were once damaged and weren't functioning that well. You can also use these to record neural function. The problem with this and all devices is scar tissue. You have a different kind of scar tissue in your brain, but it is not conductive. So if scar tissue grows around your neural probe, there's no way you can measure anything. There's no way you can stimulate anything. So on the next slide, you can see we're using carbon nanotubes. Lightweight, strong, conductive. I actually love carbon nanotubes. There are a lot of people in this field who think that they might have a toxicity problem. Our own studies basically highlight as long as you remove the catalyst used to make carbon nanotubes, and as long as they're fully pure, we're not seeing toxicity issues. I don't know if it's just us or what, but I believe there is a way we can make carbon nanotubes to be safe and used in the body. So we have seen through in vitro work, making carbon nanotubes of different surface energies reduce functions of astrocytes. Astrocytes in the brain create scar tissue, or they can be used to increase functions of neurons. On the next slide, you can see where we're taking this to an animal model. So I collaborated with Young-Yoon Lee from Seoul National University in Seoul, South Korea, obviously. She is an expert in stem cells. Do you guys remember when stem cells were supposed to be the solution to all of our problems? We're still here with problems. The problem with stem cells, in my mind, is when you inject them in the body to heal tissue or to do whatever you want it to do, those stem cells like to migrate towards healthy tissue. They don't like to stay where damaged tissue is. And that makes sense because healthy cells are producing growth factors, cytokines. Come on, come this way, right? Dead cells aren't producing anything. So they basically are favoring to go away from the area where you need them to stay. So we had a conversation. We basically said, what if we inject stem cells? These are subventricular zone stem cells from the brain with carbon nanofibers to anchor them, to keep them where stroke damage occurred. And this was a study we followed. This is middle cerebral arterial occlusion in rats. We stop the blood flow, create stroke-like conditions, inject stem cells with the carbon nanotubes, and then after 1 to 8 weeks, we basically look to see what happened. So if you go to the next slide, you can see how easy it is to visualize carbon nanotubes in the body. They're black. So whether they're hydrophobic or hydrophilic, they stayed where we injected them. They did not move around the brain, which we anticipated. that they would not move around the brain. So the next slide shows you the cells. So again, we injected them into C. This is where our imaging told us the stroke damage was. We don't see any cells in B or D. We only see the cells where the carbon nanotubes were because they're attached to the carbon nanotubes. So we were able to keep the stem cells where the stroke damage was. This is some esoteric immunohistochemistry, basically showing you that the cells, stem cells, differentiated into neurons, not glial scar tissue forming cells. So on the next slide, you can see the big thing, functional recovery. So next slide, we put the animals, the rats, on a treadmill, measured how many steps they took, and you can see that the stem cells with carbon nanofibers, more steps than stem cells alone, which of course is more than stroke alone. On the next slide, this is reflex. It's not only important for stroke victims to return motor function, but reflex pathways are important too. So in rodents, if you tickle their whiskers, their paws come out, and you can measure that spreading of their paw. Wouldn't that be great if humans did that too? So you can see also reflex functions returned faster when using the combination therapy. The last functional assay we did is on the next slide. Rotorod, how long does the rat stay on the rod? More time when using the combination therapy. Okay, so I should probably give one last example, maybe. Can you go to, do we talk about AI and sensors? Can you go to the next slide? Okay, we'll leave with this, and I'll talk about AI as we talk about this. Can you play this video? So we started a company called Quarkson, and Quarkson is a breathalyzer company. During COVID, we and many people knew that if you were positive for COVID, you had SARS-CoV-2 in your breath. Turns out you can learn a lot from your health, about your health, from your breath. So we developed a breathalyzer where we create a graphene, nanographene membrane, and we molecularly imprint it with the molecule in your breath that we want to measure. So molecular imprinting is where you just take a graphene PDMS membrane and you press the molecule onto that membrane so it creates a negative imprint so that if the molecule is present in your breath, it will attach. And when it attaches to that cavity, your resistance goes up because graphene's conductive. So it now resistance goes up and you now have a positive signal that this occurred. So we are doing this not just for COVID. In the investor world, unfortunately, nobody is investing in COVID anymore. So we've stopped working on COVID, but I'll show you where we've started moving into. So the next slide, I think, is our COVID. Yeah, if you could just get past the video. And this is our COVID data, so it worked in patients in Boston. So the next slide, oxycodone, painkiller. I never thought I'd be working with the police on research, but I am. So hopefully that helps me out in the end. So as you know, we have a breathalyzer for alcohol, but we do not have a breathalyzer for if somebody is on painkillers, too many painkillers that hampers their driving. So there's a molecule in your breath that indicates if you have had too many painkillers, too much oxycodone. We tested this with rats, with human body fluid. You don't only have to breathe into this, you can use fluid like urine as well as blood to determine this too. And again, you can see how the resistance changes when this molecule is present. The next slide is also pretty interesting. If you have early onset Alzheimer's disease, there is a molecule in your breath to indicate that. So we've created the molecular imprint of that molecule. People breathe into this, and we've started testing it on humans, which again is showing the ability to indicate if somebody has early onset of Alzheimer's. We, of course, don't quite yet know what happens in these patients. We have to wait to see if they actually get Alzheimer's disease. But in some of these patients, we do know there's a family history, and there's reasons to think through testing and otherwise that they might have early onset of Alzheimer's disease. So think about how wonderful that would be, have a breathalyzer to indicate these diseases. So I think I'm going to skip past this one. This is implantable sensors. So if you just keep on going, and I'll tell you when to stop. We're creating sensors for orthopedic implants. Keep on going. I want to stop on an AI slide and then, oh, so I need to talk about this too. So sorry, real quick. 4D printing. So we're creating materials with graphene nanoparticles that change shape when you use near infrared. If anybody has not been paying attention to near infrared, pay attention. It is a wonderful wavelength. In fact, outside of Boston, people are getting saunas in their backyards that are near infrared saunas. So you go in, you sit down like a normal sauna, and there's near infrared light all around you. Near infrared has been shown in real scientific studies, this isn't pseudoscience, to increase blood flow. So it helps with arthritis pain. It helps if you have COVID symptoms like congestion in your lungs, et cetera. So we're basically just developing an implant that's the near infrared light source. You can see the pink kind of reflecting off of the bottom. That material can change shape after we implant it. So we put it small shape in between these discs, use near infrared, it expands and separates those discs. Maybe in 5 years, there'll be more osteoporosis in that patient. The discs start collapsing again. We reignite it with near infrared and it opens up again. So there's lots of wonderful applications of what we call 4D printed materials, where the fourth dimension is time, changing shape of a material with time after it's in the body. We're also looking at scoliosis and some other diseases where this could be beneficial. So next slide. So I'd love to collaborate on that if you want. So this is the artificial intelligence part. We call it the Band-Aid. So if you go to the next slide, I can just real quickly tell you how we are incorporating artificial intelligence into biomaterials. So if you remember in the beginning, I talked about how our medicine needs to be more personalized. Well, I firmly believe that also for medical devices. We should not be implanting the same medical device in every person, the same hip implant, the same vascular stent, whatever it is, it needs to be personalized. And again, that's because we each have different immune systems. So our reaction to the same material will be different. So for example, I'm 52 years old. I don't really have any health problems that I know of. I may have a good immune system. I hope I do. So if there's bacteria on my hip implant, maybe I'm okay and my body will fight it and everything will be fine. Maybe there's a 75-year-old elderly woman who has diabetes, who has other kinds of immune system problems. Maybe they need a more active, a more aggressive nanotextured surface to kill that bacteria because their immune cell won't be able to. So we are trying to use AI, your patient history. What have you had infections? Do you have a poor immune system? Do you have diabetes? Have you had cancer? Use that information to develop an implant surface so that we ensure it's successful. That's one way we're using computer algorithms and artificial intelligence. The other way is through these sensors. So these sensors, whether it's the breathalyzer where it can gather real-time information or the one that's implanted in the body, we can know in real time what's happening. So we can also use your history, patient history, to know, is that an event that will end up with implant failure? And do we need to intervene? So I think there's a lot of opportunity to use computer algorithms in the context of your history, your patient history, to develop, design implants that will not fail. And this is just one area many researchers are looking at to incorporate AI into medicine. Okay? So hopefully that made sense. You know, this is I love this question. So I just bought an electric car and, you know, now I'm finding out we're losing all of our power charging stations in the US. So I don't know how we're going to charge our car. Save that topic for another day. But my car has over 500 sensors to be fuel efficient, well, electricity efficient, to be safe. 500 sensors. the thousands of hip implants that will be inserted in Mexico today, zero. We have fallen so far behind in medicine compared to where other fields are. So we need to start incorporating these things. And I hope you think of projects along these lines that help us get to these goals. So I think I've summarized things, I hope, well enough. Next slide. Need to thank all the funding sources, especially NIH, NSF, for giving us funds to commercialize these technologies to get into humans. The next slide, the most important slide of the talk, people in my lab group. This is Cheesecake Factory, my favorite place to go, who do all the hard work. And then the last slide is another thumbs up, but if you press on this and play it, it's cardiomyocytes in the graphene heart patch that we talked about before. So please email me. Let's improve medicine. We have a long way to go. And I thank you very much for the invitation. I look forward to coming back, talking more about collaborations and what we can do in this health area. Thank you very much. Not too bad.
Thank you, Tom. Thank you for your very nice talk. And if you have any question, please. Yeah.
Thank you, Dr. Thomas, for presenting. I have a little kind of issues with something that you've been talking about the presentation. Two things about it is what makes us say something is a failure, because about the sensor you're talking about. And about that is, I've been thinking about the range limitation, about if something is capable of measure from 0 to 10, we can say it is accepted, but beyond that range is a failure. So I've been thinking about something that we extend the range, we can improve that detection limit. So something has been taken in. The other thing about I had some issues is about what you said from the 19th. Nanomaterials is something not usual to talk, but now is very casualty. So I've been thinking about speaking the phantom material or even beyond quantum material. I don't know, is something capable of doing now. So with that quantum material, we can just extend that limitation range and something that we can improve in healthcare. I don't know, something do you have to talk about or think?
Absolutely. So thank you for the questions. I'll start. Technology is, and then incorporating that into medicine would be pico medicine. So I think you're right. to think about, let's keep on figuring out how to control materials to give us desirable properties at a smaller scale. The first question is also an excellent question. So I would say clinically, and this is maybe unfortunate, we term failure as an implant that has to be removed. So in orthopedics, if it gets infected,
Thank you, Doctor. For one of your answers, I've been thinking about when you're thinking about what you say, the pico materials. So we can see we go beyond and beyond. So when we reach the limit of the material, when we can say this is enough.
Here in this, when you were discussing about the stents, is there any possibility that we can do the vascular endothelialization or vascularization on the surface beforehand, and then inserting it into the human body? Is there any?
Absolutely.
Uh-huh.
Absolutely. And I would say I didn't have time to talk about any of this, but not only is that possible, but we have seen when endothelial cells, or other cells for that matter, attach to a nanotextured surface, they are attaching in unique receptors that is promoting their growth and the synthesis of anti-inflammatory proteins, the synthesis of pro-healing cytokines. So it's a great idea, because not only could you experimentally get a faster coverage of a stem, but those cells are more active in a good way when they're on that nanotexture surface.
Yeah.
We have not had that approach, mostly because the FDA would be highly critical of doing that. But I think it's a wonderful idea, because we know when cells attach to certain nanotextures, they're more proactive, they're healthier, they're better for that particular tissue.
Yeah, thank you. And the second question is, when you mentioned about the AI and personalized implant approach, I just wanted to ask you about this, that how when you say the usage of AI here is in terms of applying some medicines on the surface of the implants, or are you expecting that which we are creating, those textures are also can also be more personalized? Are you mentioning that?
The latter. So, I'm not a huge proponent
Yeah.
of pharmaceutical agents.
Yeah.
I'd much rather develop a nanoparticle that has nanotextures to give you a proactive response for these implants. So I'm talking more about what is the best nanotexture for that patient. And I think we actually started this about a couple of months ago. We are taking blood from immunocompromised patients, from elderly women, from the young, football star. We're taking drops of blood and putting it on different nanotextured surfaces to see how the proteins in those blood interact differently with the surface, or the cells that are in the blood interact differently. So I believe there is an optimal surface energy depending on your current health state. And again, if you don't have an active immune system for whatever reason, maybe the surface energy needs to be different.
Yeah.
Maybe it needs to be higher, maybe it needs to be lower, but the nanotexture should be different in those patients.
Yeah, and I totally agree with your point that the nanoparticles, they don't have any they don't have toxic effects on the cells. And we have been also studying 10 or 12 different, including nanotubes and everything. And we could find that the results are really amazing. But there are few patients or few people, they are allergic to some kind of nanoparticles. And that is the only thing. The numbers are less, but yes, there are some existence. So, yeah.
Like being allergic to nickel.
Yes, exactly.
Being allergic to silver.
Even copper, yeah.
Right?
Yeah.
I mean, you're absolutely right. And the thing we've always tried to do is, try to be a little independent of chemistry, but more dependent on nano.
Yeah.
That's why when we did those 3 medical devices in humans, we purposefully chose implants that were different chemistries. Because if somebody had an allergic reaction to titanium, well, there's silicon nitride, right? There's a calcium phosphate coating. So I completely agree, and that allergic response can be worse because you have more surface area of the chemistry the patient is allergic to. So, change chemistries, right? Do use a different one, but keep the nano part, and perhaps have a nice functioning drug delivery device or implant surface.
Thank you. Thanks a lot, sir. Thank you.
Thank you very much for your talk.
Sure.
You mentioned that you prefer to be chief in science rather than CEO. However, how do you identify when an idea or a thesis, or what kind of seed you identify to become a startup? And then which could be the pathway? Who are the other actors that they can be involved to be a successful company? If you can mention an example, for example.
This is a great question and a complicated question. So our first company, my first company, that I formed at Purdue, in academia at the time, and this still might be true today, was not really supportive of starting companies. I made a joke that some of the senior professors had tenure said to me, "Don't waste your time. If you start a company, that does not help you get tenure." When I was department chair at Northeastern, we actually included on our annual reviews a section on patents, starting companies and commercial. So we started to include it in annual merit reviews. But what happened at Purdue? Purdue was an excellent university to start a company because they had a technology transfer office that was proactive and huge. So I was in biomedical engineering at Purdue. They had 2 people assigned to biomedical engineering to constantly call us up, meet with us. What are you doing in the lab? What can we file for a patent on? What can we, invite you to a poster session to talk about what you're doing to investors? They were just on top of it, and that was incredibly exciting for me. I loved the fact, as I'm sure most people do, somebody liked what I was doing, right? As a 2-year, 3-year assistant professor, to know that the tech transfer office was interested in what I was doing, that was awesome. Of course, I ended up moving for them. But I think that once we filed for patents, then the technology transfer office looked for investors and basically looked around the medical device community, found an investor who I worked with. The investor was not the CEO. They did not want to be the CEO, but they wanted to give money. So then our next job, we had the technology, we thought it worked, we had funding, we had to find a CEO. So we interviewed 10, 15 people and found somebody who really gelled with us, hired them as the CEO, and then really we were off to the races. Now I understand not all universities have good technology transfer offices. Not all universities have technology transfer offices. So there are other ways you can do this, right? So when I was at Brown, I disclosed an idea about an implantable sensor. The first time when I was at Brown. I'm there now. So they thought the idea stunk. This was in 2015. They said there's no way implantable sensors are going to be approved by the FDA. Nobody is interested in having something in their body that you can read off your cell phone how you're doing. And I was like, okay, fine. What you can do in the US, and I don't know here, you can have the university give the technology to you. So it's no longer owned by the university, and you independently can start a company, all legal, everybody knows about it, it's all upfront, and I did that with this sensor technology. I went out on my own and found an investor. I went out on my own and found a CEO. It's a longer, slower process. It takes more work, but it ended up in the end, we now have a company called OrthoTag, which they're not in human clinical trials. They're trying to get there for a chip on an implant that stores your medical device records. And you can take your cell phone and download your medical device records. So you don't have to worry next time you go into a hospital or emergency room, does the clinician know about my health? Because they can read it. It's in you. So there's different models. I would say I'm the kind of person that wants to start a company on everything I do. So I have no filter, which is a problem. I wish I did have a filter. But if somebody said to me, I love what you're doing in stroke, I want to start a company around that, I'd say, let's do it. Nobody, that has not happened. I think unfortunately, as an aside, I think neurological diseases are very hard to start a company compared to orthopedics. And a lot of that has to do with commercial potential and ability to make money off of orthopedics compared to neurology. So I hope that answers your question. There is actually, I have a talk, maybe if I come back, I have a talk of 4 ways you can start a company and get regulatory approval for your technology. One is the one I mostly followed. You go through a university, what I described for Purdue. A second one is doing it on your own. A third one is really fascinating. I don't know if there are educational programs like this, but at Brown, we started a master's in engineering entrepreneurship. And that master's program, a lot of schools have this, but what makes it unique is the students have to find a research project on campus that is not commercialized and build a business plan, raise money, and then at the end of their master's, they basically present in front of investors to try to get funding. That is the real value, is there's a real, it's not just learning how to start a company, you're doing it. And so that's the third approach. I don't even remember the fourth approach. But there are great educational programs, to explore that.
Thank you. Yeah, there are many colleagues and students that maybe are interested to take these lectures or this workshop, because I think we have many ideas that can be common in startup, especially in the health
And it's a great
area.
question, because in my experience, one model does not fit everything I've done. And you have to be adaptable. You also, as the scientist, have to be willing if the CEO decides to go a different direction. And that's happened in my career, where the CEO thought, just as an example, the diabetes example. Maybe a CEO would think, oh, that's too competitive. There's too many companies doing cell encapsulation, but I want to use that for delivering stem cells to the heart. And you have to be okay with that, because if you're not, that CEO leaves. And then you have to start over looking for a new CEO, right? So I think you have to be flexible, and you have to understand there are many different roads that can get you to the end goal of getting your projects to help human health.
Thank you very much.
Yes, thank you.
Any more questions?
I'm just curious, when you were talking initially about all the metal surface modification, right, about this anodization, and you were, sort of stating that the only thing you're changing is the roughness, not the chemistry. So isn't there any change, let's say, when you do the anodization, you have these oxides, right, that are forming on top. So can you control just the roughness without affecting like the composition of the actual oxide layer, like different phases or something? And for example, when you show I think the comparison with the stents, it was, I think, when you were showing like the bare, sort of metal, is that really just bare metal and not like anodized for passivation, as you said? So how much actually are you changing independently the roughness from, to some degree, the chemistry?
Yeah, and I'm glad you were not on our FDA approval. It depends how closely you look, right? So we were required, and this is a good thing, that the FDA required us to do things like energy dispersive spectroscopy, X-ray photoelectron spectroscopy, of course, surface roughness through atomic force microscopy. The FDA, this is another interesting thing, will not allow you to use nano in the product label, and we are clearly using nano in our product label, unless you identify the novelty of the nanometer size. So you have to do experiments that show when you change that dimension, in our case, you get a different response. You have to prove the nano. You can't just make a nanomaterial and compare it to what's currently implanted. You actually have to change the nano part. So when we looked at EDS, X-ray diffraction, of course we did X-ray diffraction as well, XPS. We, at that size scale, do not see a change in crystallinity. We do not see a change in stoichiometry of oxygen to titanium. We don't see anything. But we've never done this, and probably on purpose we haven't done. We've never looked at the grain boundaries, where the grain boundaries are the high energy spot of the material. That's where all the excitement is. It is certainly possible that at a grain boundary, we have some other TI oxygen chemistry, or we have some alteration in crystallinity. You know, our implants are 60% anatase, 40% rutile, which is good because anatase is a crystalline phase that reduces bacteria growth, and you have more surface area of that. So that's part of this too. So yeah, it's a great question, and kind of the answer is it depends how closely you look. But the other thing that leads me to believe that this is chemistry independent is we have just seen the same roughness on silicon nitride, on calcium phosphate in humans in orthopedics, right? So we keep seeing the same roughness come up as optimal for these orthopedic and vascular applications. So that tends me to believe that there's some chemistry independence too. But it's a great question. And I was really surprised when the FDA gave us the report back of questions we had to answer, and they actually said only EDX. So if you've used energy dispersive spectroscopy, it is not very sensitive. And this is what the major medical approval organization in the US said use X-ray photoelectron spectroscopy, XPS, much more sensitive. If I'm on that FDA board, I'm saying XPS, not EDS. But I think they understand not every company has access to XPS, and they can do these very detailed chemical analysis.
I have a question. Nanotexture materials can perform different wettability behaviors, and with the change of the roughness factor, what is your recommendations for this kind of successful implants to perform a hydrophobic performance or hydrophilic performance? For example, hydrophobic for the antibacterial performance, but also the hydrophilic for the biocompatibility with the cells in the human body.
It sounds like a contradiction, right?
Yeah.
How can I both create hydrophobic and hydrophilic surface? So I can say in general, I have moved beyond categorizing things as hydrophobic or hydrophilic. What I like to do is categorize the specific surface energy. Right, so if in this example where we see 42.5 millinewtons per meter is the ideal surface energy, that is certainly not hydrophobic. However, there are higher surface energies, so it's not as hydrophilic as it could be, right? We've tested some materials that people create with surface energy is upwards of 70, 80. So we certainly are in between both hydrophobic and hydrophilic. And again, I think that the reason for that is a couple of proteins that reduce bacteria, reduce inflammation, promote bone growth they just happen to be around 42.5 millinewtons per meter. So it's interesting to think about that, and why that is the case. But we can target one surface energy to get proteins that can do all of these things. Now, the other thing I can say is these are all general trends, right? When we create an implant surface that's 42.5 millinewtons per meter by anodizing titanium to have a 60 nanometer roughness, we are increasing mucin adsorption, vitronectin adsorption. We are not getting exclusive adsorption. Right? There are still many proteins on that surface. We've just increased it, by 5%, 10% of the surface is now more mucin, more vitronectin, and that seems to be enough. So, yeah, it's a great question. The other thing that is a great question that we haven't really figured out how to do the bioactivity of these proteins, once they absorb to a nanotextured surface, is much different than a smooth surface. So we looked at, there were a couple of questions about sense. We looked at and published a paper on fibrinogen absorption. And we created a surface that inhibited fibrinogen absorption. We still got some to absorb by changing nanotexture, but the fibrinogen bioactivity was much different on the nanotextured surface. It was absorbing almost like a sphere, a non-bioactive form. On the non-nano surface, the fibrinogen was absorbing in a fibrillar form, which is reported to be the more bioactive form. Fibrinogen promotes platelet attachment and thrombus formation and all of this stuff. So bioactivity is an important part that should be one of our next goals. Can we create an equation that promotes bioactivity of the proteins that we're enhancing absorption?
Okay.
Very long answer to a good question.
No, thank you very much.
Last question? No? Okay. Thank you, Dom, for your very nice talk. We have a recognize.
Is this a certificate? Awesome.
Yeah.
I don't know how
Sure.
to turn it off.
And I have a little present to you.
Excellent. Oh.
I think you will be fine.
Very nice. Thank you very much.
Thank you.
Thank you.